So now let's look into the microbial isolate whole genome sequencing application on the nanopore sequencing platform. So for this microbial whole genome sequencing, we have developed something called a NOMIS protocol. So it actually stands for nanopore only microbial isolate sequencing solutions. So with this approach, you can generate complete high quality genome and effectively resolve the plasmids as well. And additionally, it also allows for accurate profiling of antimicrobial resistant genes. It offers you the informations into the resistant mechanism. And this method is very highly flexible. It can support sample batching. It, or if let's say you don't require any batching, you can do that as well. And it also can deliver, it also can deliver rapid turnaround times, making it very ideal for both clinical and research setting, where speed and adaptability are essential. So I think we mentioned it earlier that one of the major benefits of um, nanopore sequencing is the ability to generate a very long DNA reads, while the short read sequencing technology can give you hundreds of base pair long. Nanopore sequencing technology give you reads that are tens of thousands of base pair long. So this can come in handy when you are looking at the resistant genes, particularly if you want to look at or if you want to know something about where in the bacterial genome that the resistant gene is. And also when you sequence using short read sequencing technology, it can be hard to get this information because bacterial genome have a lot of repetitive elements, and short read sometimes is simply not long enough to cover these repetitive regions. So even if you sequence the whole genome, if you try to stitch the reads together and assemble them back into the original genome sequence, you also may end up with a bunch of short stretches of sequence, and you don't know how exactly where that fits. Where with long reads you can actually um, sorry. So with long read, you can actually, span this repetitive region, then you're actually able to recover a full circular sequence of the bacterial sequence as well as a plasmid. So instead of like, for example, knowing a bacteria carry a certain resistant gene, you can know like where in the genome that gene is. So this is the overview of the end-to-end microbial isolate whole genome sequencing protocol. So this is a very flexible approach that allows you to sequence up to 24 microbial isolate genome per mean ion flow cells and generating a minimum coverage of 50x per genomes. And this 50x coverage threshold is what we tested and validated that is sufficient for downstream analysis, including accurate assembly, and accurate plasmid resolution, AMR profiling, as well as the SNP genotypings. And you can also get access to analyze your sequencing data using our software Epitomy, which is a very user-friendly bioinformatics tool. So let's look at the sample preparations. So in terms of the sample preparations, we have developed four optimized extraction methods to generate high quality DNA from your cell cultures. So it really depends on whether you are looking into doing like a universal applications on bacteria, fungi, or yeast. Then you can follow this extraction method to get a average sequencing read length of 4 to 7 kb. However, if your research require more than 7 kb, then it depending on the sample you are working on, whether it's a bacteria or is it very hard to lyse organism or is it fungi. We also have the optimized the protocol, and you can follow this protocol accordingly to get the high quality genomic DNA for sequencing. And on the right hand side, you can see here that we have already listed here the extraction kits that we use to optimize the protocol, as well as the sample input requirement, the expected yield, as well as the expected DNA integrity number. So for sample extraction QC in general, we typically use two methods for DNA quantifications, the nano drop as well as qubit. So nano drop, as we know, is a spectrophotometer that measure the DNA concentration based on the absorptions at 260 nanometer. This is very widely used because it's very quick, it's very easy, and it also provides both the concentration and purity of your samples. And it also measure important ratio like 260 to 80 or 260 to 30, which can indicate the possible contamination with protein or salts. And for qubits, this is different concept. It is a fluorometer, so it uses a fluorescent to measure the DNA concentration and is much more specific. So it quantifies the DNA by binding to a dye that only fluoresces when it binds to double-stranded DNA, so making it highly accurate for determine the actual amount of DNA that actually present in your sample. So in sequencing, with nanopore sequencing including, it's very important to use nano drop reading together with the qubit measurement for more accurate quantifications. So this is because nanopore reading can sometimes overestimate DNA concentration, especially if there are contaminants like RNA, phenols, or other chemicals that left over or carry over from the extraction process. So if you observe a very significant difference between nanodrop and qubit readings, this could be a red flag for you to, investigate whether your sample contain impurities that may affect the downstream process. So, in general, the pure DNA should give you, A260 to 80 ratio of about 1.8 and 260 to 30 of about 2 to 2.2. And if let's say you get a much lower ratio, it also can indicate phenol contamination. But it also can indicate carryover of guanine or carbohydrates. And for long read sequencing technologies such as those, for example, like including Oxford Nanopore, getting a long DNA fragment is essential. The longer your DNA fragment with, especially in nanopore, we have no limits on how long is your DNA fragments. So the better the read length and the more accurate the assembly is, and it enables the structural variance detections. And there are a variety of methods available to do fragment analysis. For example, the gel-based analysis, or you can use bioanalyzer or tap stations or femtopulse. However, if you are expecting fragments larger than 20 kb, then femtopulse would be would be ideal. So let's go into the library preparation part. So when you get your DNA ready, right? In this protocol we recommend 200 nanogram of extracted genomic DNA. With this method, it has developed the process up to 24 samples with a genome size of up to 7 MB simultaneously. So if you are working on a genome size that is like smaller or larger than 7 MB, then you can adjust the multiplexing accordingly, depending on, of course, a number of factors like, for example, the sample quality, your genome size, or the coverage requirements. So on the right-hand side of the slide, right, you can see that this is the overview of the library preparation process. And the process begins with the segmentation step, where the transposase will simultaneously fragment and then add barcodes to the end of the DNA in one step. So this particular step only takes 15 minutes. And then after that, the barcoded libraries are then pulled, followed by impure bits cleanup, then attachment of the sequencing adapter to the DNA ends. And this entire process only take 30 minutes. So once the library is prepared, you can then prime your floor cell, load the library for the sequencing. So as you can see, the entire library preparation process is so quick, so easy, and so straightforward that gets you ready to start sequencing in less than 60 minutes. So analysis. So you can sequence up to 72 hours. However, it really also depending on, for example, the number of samples that you want to sequence at a time. Because if you sequence just a few samples, then you don't need to let it sequence for 72 hours. You can maybe just let it sequence for 8 hours or for 24 hours. As long as you generate enough data, then you can stop the sequencing, watch the flow cell, keep it for your next run. So it allows you this flexibility with Nanopore. And for sequencing software and data analysis, we have Mino, a very user-friendly operating software that drive nanopore sequencing devices, like for example, including data acquisition, real-time analysis and monitoring, local-based calling, data streaming. It also allows you to set different running parameters according to your needs as well as applications. And for data analysis, we have Epitome, um, Epitome have many analysis workflow that you can use. And it can be run either on the graphical user interface or command line. And these are free workflow, so you don't need to pay for the use of this workflow. Like for example, for bacteria whole genome sequencing, you can use WF bacteria genomes for you to do bacterial assembly, the annotations, the MLST, as well as allows you to predict the antimicrobial resistant profile as well.
So so it's free of charge and also include how many CPU and RAM are only available to download, or we can run it in cloud, or we have to pay for cloud.
Mm. So you can run it locally. You can also run it in cloud. Both are free. Mm.
Okay, okay. So it means that the computation on using the CPU and RAM also free and storage also free.
Correct.
Okay, perfect. Thank you.
Yeah. So the Epitome platform, as we mentioned, is really a user-friendly workflow because we know the importance of having, the user-friendly workflow for scientists to streamline the analysis of the bacterial genomes. I'm sorry, I'm not sure why is it keep going to the next slide. So these two, it allows for multiple features. This workflow it allows for multiple features that simplify the process of characterizing the bacteria sample, making it accessible even if especially some wet lab scientists that don't have intensive bioinformatics experience. We also have integrates the multi-locus sequence typing MLST, a widely used method for strain identification and epidemiology tracking. So this workflow automatically identify the bacterial species and provides a profile for eh, any species just now I'm here, right? Yeah. It allows any species with the publicly available MLST profile. And here, this one is the antimicrobial resistant profiling. So this workflow also allows the identifications of both acquired resistant genes as well as the chromosomal mutation that mediates the AMR. And also, if you are working with or if you are working on the food safety, and if you are looking for a Salmonella serotyping prediction tool, feel free to reach out to us. We have the end-to-end solution using the FDA approved 602, which allows for precise stereotyping. And it also provides the prediction for subspecies and the all H1, and H2 antigens. And these predictions are important for identifying for specific serotypes of Salmonella, which is critical factor in tracking foodborne outbreaks and ensuring the public health safety. So this here is a case study demonstrate the power of whole genome sequencing for outbreak investigation, as well as the antimicrobial resistant prediction in a clinical microbiology settings. So the author is from the Providence Health Care Labs in Canada. They apply nanopore long-read sequencing to investigate a cluster of multi-drug resistant Shigella shonei and aim to genetically characterize the MR genes and support potential interventions. So what they do is that they have sequenced 56 isolates using only the nanopore platform, and they have found a strong evidence of a clonal strain responsible for the infections. And also the key highlights here is that from the study is that there was a very high concordance between a phenotypic antimicrobial susceptibility and the genotypic prediction generated by the nanopore sequencing. So this included the accurate detection of the SNP that confer resistance to the quinolones, a very critical antimicrobial drug class. And also this analysis has showcased the base level accuracy of nanopore sequencing, particularly in predicting the antimicrobial resistance directly from the genomic data. So I think this is really a valuable um, work that showcase, in a outbreak scenario, nanopore sequencing can allow for a rapid intervention and better informs clinical decisions. And here in this slide, this is a very recent blog from Dr. Ryan Wick from Australia. So he has done like a benchmarking using our new base calling model for nanopore sequencing. And you can see that based on his benchmarking results, you can see that there is this significant improvement in read accuracy and even greater improvement in consensus accuracy. And this includes, in this list, some of the, like for instance, the E. coli or Salmonella enterica that has about 50 to 53% of GC content. And you can see that from his comment here, he said that, this is the biggest improvement that I've seen in a while. Most of these ONT only bacterial genome are now more than Q60.